going to be talking about the development of the reproductive system. I think for this topic, it would be best to divide it into the development of the gonads and the duct system in male and females, followed by the development of the external genitalia, and then finally the descent of the testes and to some degree the ovaries as well. So to start off with some basic general embryology, we know that our embryo consists of a trilaminar disc consisting of three layers. You have the ectoderm, the mesoderm, and the endoderm. And what we know is that the urogenital system is derived from the mesoderm. The mesoderm is going to have three different types depending on its orientation within the embryo. So you could have the paraxial mesoderm, the intermediate mesoderm, or the lateral plate mesoderm. Our urogenital system is derived from the intermediate mesoderm, and it predominantly gives rise to the urinary system. And to get the reproductive system, we're mainly concerned with the mesonephros, which gives rise to the gonads. Starting with our basic indifferent gonads. So essentially, till the 4th week of gestation, we don't know whether the embryo is male or female. So we cannot distinguish whether the embryo is dimorphically male or female. And in order for the gonads to develop and form into a distinguishable male or female, we require that our primordial germ cells migrate from the yolk sac through the vitelline duct, or for the sake of simplicity, we could also say the dorsal mesentery of the hindgut to the urogenital ridge. The urogenital ridge is basically just a proliferation of surface epithelium and mesenchymal cells. And with the primordial germ cells finally infiltrating the gonadal ridge, we are able to form the gonads. Without this, you wouldn't be able to form gonads. So we can say that the primordial germ cells have somewhat of an inductive effect on the formation of the male and female genital systems. During and before the infiltration of the primordial germ cells to the gonadal ridge, we have the formation of something called the primitive sex cords within the urogenital ridge. And during about the 4th week and after, these primitive sex cords are going to eventually develop into our male or female medullary and cortical cords. But before we get to the specifics, we have to familiarize ourselves with the different ducts that we see in the development of the male and female reproductive system, starting with the mesonephric duct in blue, which is what gives rise to the male reproductive or male urogenital system. And then you have your Mullerian or paramesonephric ducts in pink that give rise to the female genital system. And in yellow, you have the gubernaculum, which we talk about primarily while discussing the descent of the testes. And you can see that they're all sort of inserting into the urogenital sinus, which goes towards the cloaca. Let's talk more specifically about the testes and the male duct system. So we have to cover a little bit of molecular regulation here. So we know that a male embryo is going to have an XY chromosome combination, and essentially that Y chromosome has this gene called the SRY gene, which stands for the sex-determining region on Y. And this gene, when active, is going to secrete something called the testes-determining factor. And the testes-determining factor is then going to allow the proliferation of the primitive sex cords into medullary cords within the medulla of the testes. And gradually, these cords are going to break and form the rete testes and eventually the dense layer of fibrous tissue that covers the testes called the tunica albuginea. And by the 4th month, our testes are going to become sort of horseshoe or U-shaped, where the rete testes then are going to be able to connect with the cords within the primitive gonad. At this point, we've also formed our sustentacular cells of Sertoli and the Leydig cells in the interstitium, which we know of from histology. So we know that our Sertoli cells are going to secrete something called the anti-Mullerian hormone or the Mullerian inhibiting factor. And what the Mullerian inhibiting factor does is that it inhibits the growth and proliferation or differentiation of the paramesonephric or the Mullerian ducts that we need to see differentiated in order for a dimorphically female individual to form. So the anti-Mullerian hormone or the Mullerian inhibiting factor that's secreted by the Sertoli cells will prevent the paramesonephric ducts from fully differentiating. Meanwhile, the Leydig cells are going to secrete testosterone, which is going to stimulate the processes that result in the proliferation and growth of the mesonephric ducts into the male reproductive organs. And also, the testosterone is acted upon by an enzyme called 5-alpha reductase, which then converts testosterone into dihydrotestosterone that allows for the differentiation of the external male genitalia, which we will talk about later. So, talking about the male ductal system, we know that the testes remain solid until puberty. And upon hitting puberty, the duct system will acquire a lumen, which then is called the seminiferous tubules. The seminiferous tubules will join the rete testes, which then enter the ductile efferents. And the rest of the mesonephric duct is going to sort of form into what we know as the vas deferens. Apart from that, we also know about the epigenital tubules and the paragenital tubules. The epigenital would join with the rete testes and the paragenital that do not. And as the mesonephric duct continues to grow, at the entry of the efferent ductules, you have a huge convolution of the mesonephric duct, which gives rise to our epididymis. And as it continues to grow and differentiate, we also see a sort of outbudding out of the mesonephric duct called the seminal vesicles. And you also have the formation of your ejaculatory duct. Because of the secretion of the Mullerian inhibiting factor, your Mullerian ducts or your paramesonephric ducts are going to regress apart from a tiny vestigial part that we then know as our appendix testes. So that's about it for the male gonads and duct system. Now we talk about the female gonads and duct system. So we know from the molecular regulation that we just discussed that with a female, you'd have an XX chromosomal combination. So no Y chromosomes, no SRY gene, no testes determining factor. So no Mullerian inhibiting factor, no testosterone, which means that our paramesonephric ducts are going to continue to grow and proliferate and differentiate. But because there's no testosterone, instead our Wolffian ducts or our mesonephric ducts are going to regress. So coming to the female genital ducts, we mainly talk about the effect of estrogen and the lack of testosterone and Mullerian inhibiting factor on the growth and development of the paramesonephric ducts. Looking at the diagram here, we can distinguish three different parts of the paramesonephric duct. So you have the vertical cranial part, the medial part that crosses the mesonephric duct in blue, and the caudal vertical part. The first two parts will fuse together and form the uterine tube and the uterine canal. And as on both sides, the paramesonephric duct turns medially, the paramesonephric ducts then fuse and become visible in a cross section or a transverse section, which we can see here. And as the paramesonephric ducts fuse, they form the broad ligament of the uterus. And eventually the fusion of the paramesonephric duct also gives rise to the body and the cervix of the uterus and the upper 2/3 of the vagina. Also, we can see that it also helps divide the pelvic cavity into the uterorectal part and the uterovesical pouch. The surrounding mesenchyme around the uterus is then going to give rise to the muscular myometrium and the peritoneal perimetrium. When there's a lack of testosterone due to the absence of a Y chromosome, we will also observe that the mesonephric ducts or the Wolffian ducts that are responsible for male genital duct development are going to regress. Moving on, we also talk about the vagina. So we know that the vagina is developed from two different parts. We've already seen that the upper two-thirds of the vagina is going to be developed in the same fashion that the uterus and the uterine tubes are formed. But we also see that the part of the paramesonephric duct that is in contact with the urogenital sinus is going to result in the formation of two vaginal evaginations, and these are going to grow and proliferate till they form a vaginal plate. And that vaginal plate is then going to have a canal formed through it by the 5th month of development. And then we're going to observe changes like the formation of the vaginal fornices around the mouth of the uterus and the formation of a single layer of vaginal cells that separates the vagina from the urogenital sinus, which we call the hymen. Coming to molecular regulation, we've already discussed how the presence of the Y chromosome then results in the presence of the SRY gene and how that will upregulate the SOX9 gene to then secrete steroidogenesis factors and other genes that will then result in differentiation of the Wolffian duct into the testes. At the same time, this SOX9 gene is going to downregulate WNT4. When we think about females, we don't have an SRY gene because we don't have a Y chromosome. So you'd have the WNT4 gene instead that is going to upregulate the DAX1 gene that's going to then downregulate the SOX9 gene and then inhibit all the characteristics that we would see rising from the presence of testosterone and the effect of the SOX9 gene. So the WNT4 upregulates the DAX9, which then downregulates or inhibits all the mechanisms of the male molecular regulation, which then allows us to form the ovaries from the paramesonephric duct system. Now we talk about external genitalia. Just like we saw with the gonads, we always start off with an indifferent external genitalia. And the characteristics of this indifferent external genitalia consist of a genital tubercle, a cloacal membrane, the cloacal fold, which is essentially just an aggregation of mesenchymal cells that have migrated to either side of the cloacal membrane, and the genital swellings. Later on, these cloacal folds are then going to develop to form the urethral folds anteriorly, and posteriorly you have the anal fold. In males, we see that the genital tubercle is going to elongate a bit, and as it does this, it also pulls with it the urethral folds, and eventually, the urethral folds are going to fuse and close around a urethral groove, which forms the penile urethra. But this doesn't go all the way up to the glans penis. Instead, around the fourth month of development, and an ectodermal inwards migration of cells is going to result in the formation of the external urethral meatus. Meanwhile, the scrotal swellings are going to arise from the genital swellings that we see in the indifferent external genitalia. For females, the genital tubercle is going to develop into the clitoris. The urethral folds, instead of fusing, are going to give rise to the labia minora. The urogenital groove will become the vestibule, which is where you can find the openings of the urethra and the vagina. The genital swellings become the labia majora, and that is mainly it for external genitalia of the female. Usually, and this is just a fun fact, the clitoris is actually enlarged more than the phallus in the embryo. So that is a method that doctors could use to see if the embryo is actually male or female. Now we talk about descent of testes and the ovaries, mainly the testes. So we already know that the testes are formed retroperitoneally at the posterior abdominal wall, and that they're sort of brought into the scrotum through the inguinal canal, which is about 4 centimeters long. And towards the end of the second month, there's a urogenital mesentery that attaches to the testes and the mesonephros at the posterior abdominal wall. But because the mesonephros is going to degenerate eventually, this mesentery is just some sort of ligamentous attachment for the testes. And caudally, this urogenital mesentery will become known as a caudal genital ligament. And along with it, at the caudal pole of the testes, you also have something called the gubernaculum. And this gubernaculum is going to reside between the developing external and internal abdominal oblique muscles. And as the testes descend toward the internal inguinal ring, the extra abdominal portion of the gubernaculum will form and grow from the inguinal region towards the scrotal swellings. So when the testes are passing through the inguinal canal, the extra abdominal portion of the gubernaculum contacts the scrotal floor. Usually, the timeline for the descent of testes is such that the testes will reach the inguinal region by approximately 12 weeks. They migrate through the inguinal canal by 28 weeks, and they should reach the scrotum by 33 weeks. The factors that affect the descent of testes are usually just the effect of androgens and Mullerian inhibiting substance. And because the testes are passing through the abdominal wall to reach the scrotum, the layers of the abdominal wall will also become the layers of the testes. So what I mean by that is that the transversalis fascia will eventually become the internal spermatic fascia. The internal abdominal oblique muscle will become the cremasteric fascia and muscle, and the external abdominal oblique muscle will become the external spermatic fascia. We don't really talk about descent of the ovaries too much, other than the fact that the cranial genital ligament, which is again derived from the urogenital mesentery, will form the suspensory ligament of the ovary, and the caudal genital ligament will form the ligament of the ovary proper. The round ligament of the uterus is also derived from the caudal part of the urogenital mesentery, and that will extend into the labia majora. That is pretty much it for development of the urogenital system. We also can talk a bit about the timelines with the help of this diagram, starting from 4 weeks up until birth and even after. So that's a summary of urogenital system. Thank you.